Good morning, sir. I am Ajay. My topic is on GLP and GMP. GLP, good laboratory practice. GLP is an FDA regulation. GLP is a formal regulation that was created by the FDA in 1978. GMP embodies a set of principles that provide framework within which laboratory studies are planned, performed, monitored, and are achieved and reported. Why was GLP created? In early 70s, FDA became aware of cases of poor laboratory practice all over the United States. They discovered a lot fraudulent activities and a lot of poor lab practices. Examples of some of these poor lab practices found were equipment not being calibrated to standard form, therefore giving wrong measurements, incorrect or inaccurate accounts of the actual lab study, inadequate test systems. Purpose of GLP. GLP is to certify that every step of the analysis is valid or not, assure the quality and integrity of data submitted to FDA in support of the safety of regulated products. GLP have heavy emphasis on data recording, record and specimen retention. Good laboratory practice principles. It has 10 principles: test facility, organization and personnel, quality assurance program, facilities, apparatus, material and reagents, test systems, test and reference substances, standard operating procedures, performance of the study, reporting the study results, storage and retention of records and materials. First, we take test facility, organization and personnel. Study personnel responsibilities should have the knowledge of the GLP principle, access to the study plan and appropriate SOPs, compile with the instruction of SOP, record raw data. Study personnel are responsible for the quality of their data, exercise health precaution to minimize risk, ensure the integrity of the study. Second, quality assurance program. Responsibilities of the QA personnel: access to the updated study plan and SOPs, documented verification of the compliance of study plan to the GLP principle, inspection to determine compliance of the study with GLP principle. Three type of inspection: study based inspection, facility based inspection, process based inspections. Inspection of the final reports for accurate and full description. Report the inspection results to the management statements. Third, facilities: suitable size, construction and location, adequate degree of separation of the different activities, isolation of test system and individual projects to protect from biological hazards, suitable room for diagnosis, treatment and control of disease, storage rooms. Apparatus, material and reagent, apparatus of appropriate design and adequate capacity, documented inspection, cleaning, maintenance and calibration of apparatus and materials not to interfere with the test system. Chemical reagents and solutions should be labeled to indicate, identify expiry and specific storage instructions. Fifth, test systems: physical and chemical test system, biological test system, record of source, date of arrival and arrival condition of test systems, proper identification of the test systems in their container or when removed, cleaning and sanitization of containers, pest control agents to be documented. Sixth, test and reference items: receipt, handling, sampling and storage, characterization, known stability of test and reference items, stability of the test items in its vehicle, experiments to determine stability in tank mixers used in the field study, samples for analytical purposes for each batch. Standard operating procedures (SOP), written procedure for a laboratory program. They define how to carry out protocol, specified activities, most often written in a chronological listing of action steps. They are written to explain how the procedures are supposed to work. Action to be taken in response to equipment failure. Performance of the study: prepare the study plan, Content of the study plan, identification of the study, records, dates, reference to test methods, information concerning the sponsor and facility, conduction of study. Reporting of study results, information on sponsor and test facility, experimental starting and completion dates, a quality assurance program statement, description of materials and test methods, results, storage, जैसे sample, reference item, raw data, final report etc. 10th, storage and retention of records and materials. The study plan, raw data, samples, inspection data and master schedule, SOP, maintenance and calibration data. If any study material is disposed of before expiry, the reason to be justified and documented. फिर index of material retain. बेनिफिट क्या है जीएलपी के? इट विल गिव बेटर इमेज ऑफ कंपनी एज ए क्वालिटी प्रोड्यूसर इन ग्लोबल मार्केट, प्रोवाइड गाइडलाइन फॉर डूइंग टेस्टिंग एंड मेजरमेंट इन डिटेल, प्रोवाइड गाइडलाइन एंड बेटर कंट्रोल फॉर मेंटेनेंस ऑफ इंस्ट्रूमेंट्स, एनवायरमेंट कंट्रोल, प्रिजर्वेशन ऑफ टेस्ट रिकॉर्ड etc. Provide hot tips on analysis of data as well as measure uncertainty and perfect record keeping. Second, GMP, good manufacturing practice. GMP is a set of regulation. Good manufacturing practice. GMP is a set of regulations, codes and guidelines for the manufacture of drug substances and drug products and medical devices, in vivo and in vitro diagnostic products and foods. जनरल रिक्वायरमेंट क्या है इसके लिए? अवॉइड रिस्क एंड पॉसिबिलिटीज ऑफ मिक्सअप एट ऑल स्टेजेस ऑफ एफएफजी, लेबलिंग, पैकेज एंड टेस्टिंग, रेगुलर मॉनिटरिंग ऑफ टेम्परेचर एंड ह्यूमिडिटी, पार्टिकल काउंट, डीओपी टेस्टिंग etc. प्रॉपर ड्रेनेज सिस्टम which prevents backflow, building and facilities, design or construct features, lighting, ventilation, air filtration, air heating and cooling, plumbing, sewage and refuse, washing and toilet facilities, sanitation, maintenance. GMP, a basic principle of GMP is that quality cannot be tested into a batch of product, but must be built into each batch of product during all stages of manufacturing process, or it is designed to minimize the risk involved in any pharmaceutical production that cannot be eliminated through testing the final product. 10 principle of GMP. Design and construct the facilities and equipment properly, follow written procedure and instruction, document work, validate work, monitor facilities and equipment, write step-by-step operating procedure and work on instructions, design, develop and demonstrate job competence, protect against contamination, control components and product-related processes, conduct planned and periodic audits. GMP category: sale, premises, equipment, personnel, sanitation, raw material testing, manufacturing control, packaging material testing, finished product testing, quality control department, records, samples, stability, sterile product. First, sale: no distributor and no improper sell a drug unless it has been fabricated, packaged, labeled, tested and stored. Premises and equipment: permits effective cleaning, prevent contamination, orderly conditions, good state of repair. Personnel: appropriate education, training and experience, sufficient number of people receive GMP training. फिर है sanitation program to prevent contamination, limit the sources and types of contamination, cleaning procedures for facilities and equipment, pest control, environment monitoring. Raw material, packaging material and finished product testing. Each lot or batch of raw material is tested. Confirm the identity of the raw material. Samples of incoming material are collected and tested before use. Approved test methods and specification are used. Results must conform to specification for release for use or sale, transportation and storage records. Manufacturing control: Written procedures are established and followed. Critical process validated. Second person verification of activities. Self-inspection program. Quality control department. Quality control responsibilities. Testing of bulk components prior to use by production. Testing of finished product prior to release for sale. Stability program. Review batch record, labels, release product based on QC test results. Training, auditing, customer complaints. Records. Document all GMP activities. Use good documentation practice GDP. Records must be readily available. Good documentation practices. Documentation must be permanent, legible, clear, concise, accurate, timely, and complete. Samples. Retain samples of each lot of raw material and finished product for specified period of time. Stability. Establish the length of time in which the product meets all specifications. Monitor the drug for this period of time. Sterile products packaged in separate and closed area by trained personnel using methods to ensure sterility. Why GMP is important? A poor quality medicine may contain toxic substances that have been unintentionally added. A medicine that contains little or none of the claimed ingredients will not have the intended therapeutic effect. Thank you.